Let's kick things off with a fundamental question, really the central paradox of virology. How can a virus with a genetic blueprint that's just ridiculously small, I mean, often thousands of times smaller than its hosts, completely hijack a complex human cell and turn it into a non-stop virus factory? Seriously, how does so little information achieve so much? Well, in this explainer, that's exactly what we're going to unpack. We're diving into the absolute masterclass in genetic efficiency that is viral gene expression. You see, for a virus, this isn't just about surviving. It's about total cellular domination through pure, ruthless optimization. So here's our game plan. First, we'll look at the massive challenge viruses are up against. Then we'll survey the cellular environment, the playing field. We'll get into how they basically tear up the rulebook of molecular biology, explore their incredible strategies for getting the most out of their tiny genomes, and then we'll finish by looking at some of the most sophisticated viral manipulators out there. All right, let's get into it. Section 1. At its core, the story of a virus is one of incredibly limited resources versus absolutely huge ambitions. They show up at one of our cells with basically nothing more than a tiny bit of genetic code and this relentless, all-consuming drive to make more of themselves. And this is the critical point. They face this dual challenge. Their teeny tiny genomes have to pull double duty. On one hand, they need to code for all the proteins to copy themselves. You know, the polymerases, the capsid proteins, the whole shebang. But at the same time, they also have to code for proteins that can disarm, control, and manipulate the host cell. That's a huge ask for such a small genetic budget. So how on earth do they manage this? Well, to understand their strategy, we first have to understand the battlefield. So, Section 2, the playing field. Every single move a virus makes is shaped by the basic geography of a eukaryotic cell, that fundamental divide between the nucleus and the cytoplasm. And this battlefield, of course, operates under a very strict set of rules, the central dogma. You know this. In any of our cells, the flow of information is tightly regulated. DNA gets transcribed to RNA inside the nucleus, and then that messenger RNA is shipped out to the cytoplasm to be translated into protein by ribosomes. This separation in both space and process is the standard way of doing things, a procedure that viruses are about to completely exploit. And this slide shows two totally different ways of navigating that cell. Over on the left, you've got your typical DNA virus. It kind of follows the host's logic. It gets its DNA into the nucleus to use all that lovely transcription machinery that's already there. But look at poliovirus on the right. It's an RNA virus, and it just completely ignores the nucleus. Its entire life cycle, translation, replication, assembly, it all happens out in the cytoplasm. It basically treats the nucleus like it doesn't even exist, which just goes to show how viruses can exploit different compartments to suit their needs. Which brings us perfectly to our third section. See, viruses aren't just playing on the field. They are actively rewriting the rules of the game. They are absolute masters at disrupting that central dogma we just mentioned. And this diagram just brilliantly shows the scale of that disruption. On the left, that's our cell, right? Familiar, predictable. But on the right, an infected cell. It's controlled chaos. Viruses introduce pathways that are normally forbidden in a cell. You see negative sense RNA, the non-coding strand, being used as a template to make messenger RNA. You see RNA being reverse transcribed back into DNA. Viruses don't just add more cars to the existing roads, they build entirely new information highways, and they all lead to one place: more virus. So how do they do it? Well, a key part of the answer lies in the machinery. Our cells use DNA-dependent RNA polymerases. They read a DNA template to make RNA, and they need a primer to get started. Many viruses, though, they just bring their own specialist tool, an RNA-dependent RNA polymerase, or RDRP. This enzyme is a complete game changer. It can read an RNA template to make more RNA, and crucially, it often doesn't even need a primer. And this enzyme is famously error-prone, which, as it turns out, is a major driver of viral evolution, leading to the rapid emergence of new strains. And when they do need a primer, they get incredibly creative. Some, like poliovirus, actually use a small protein, VPG, as a primer. Others, like Bunyava viruses, do something called cap snatching. They literally steal the 5' caps from our own messenger RNAs and stick them on their own. It's this brilliant combination of sabotage and recycling. It's utterly ruthless and just incredibly efficient. Okay, let's move on to Section 4, where we really get into how viruses solve that initial problem of having such tiny genomes. This is where we see their true genius for compressing and expressing information. So how do you pack so much function into such a small genetic space? Well, this is their playbook. We're going to focus on a few of these, but some key strategies are making single messenger RNAs that code for multiple proteins, using overlapping reading frames where one piece of DNA can be read in different ways to make different proteins, and of course, using alternative splicing to create a huge variety of proteins from just one gene. Let's dig into that first one. Okay, so let's start with this concept, polycistronic mRNA. In our cells, it's pretty much one mRNA, one protein, that's monocistronic. Viruses, they throw that rule right out the window. They make these polycistronic messages where one single long mRNA can be translated to produce a whole suite of different proteins. It's a massive space saver.
And here is that exact strategy in action. This is the poliovirus cycle again. If you look at step 4, you can see its single RNA genome being translated into one giant polyprotein. Then, in step 5, the virus's own proteases get to work, chopping up that massive protein into all the individual functional pieces it needs, the structural proteins, the polymerase, all of it. So that's one message, one translation event, but a dozen or more final products. Just brilliant.
Poliovirus also has another amazing trick up its sleeve. You see, our ribosomes normally need to see that 5' cap on an mRNA to even start translating. Poliovirus doesn't bother with one. Instead, it has what's called an iris, an internal ribosome entry site. This is a complex, folded RNA structure that basically acts as a landing pad, letting the ribosome bind directly to the middle of the message and start translating. It completely bypasses the cell's normal rules. It's a direct hijacking of our protein-making machinery. All right, for our final section, let's look at some of the most sophisticated strategies of all. Here, we're going to see viruses that don't just use the host machinery, they manipulate it with a level of complexity that is, frankly, just staggering. And our case study for this is HIV, a retrovirus. Its life cycle is already wild. It uses reverse transcriptase to turn its RNA genome into DNA, which then gets integrated permanently into our own genome. From that moment on, it is completely dependent on the host cell's own RNA processing systems to make new viruses. Now get ready for this, because this is the astonishing part. The integrated HIV genome has only about 9 genes. 9. Yet by cleverly manipulating the host cell's splicing machinery, it can generate more than 40 different messenger RNAs from that handful of genes. That is an absolutely incredible amplification of information. Just think about that. And this is how it works. HIV doesn't invent some brand new process, it just hijacks one that our cells already use all the time, alternative splicing. By using its own regulatory proteins, like TAT and REV, it can influence which exons are included or excluded from the final mRNA transcript. It basically conducts the host splicing machinery like an orchestra, telling it to produce this enormous diversity of viral products from a single genetic template. It's not just using the system, it's directing it. So the final act of manipulation is what we call host shutoff. Because it's not enough to just make more of your own stuff, you have to stop the host from making their stuff. Viruses like herpes simplex, or HSV, do this in 2 brilliant ways. First, they deploy a protein called VHS that acts like a wood chipper, just shredding cellular mRNAs to get rid of the competition. Second, they use other proteins like ICP27 to act as a bouncer. This protein specifically recruits host factors that grab the viral mRNAs, protect them, and give them a VIP escort out of the nucleus, leaving the host's own messages behind to rot. So, as we've seen, viruses are the ultimate minimalists. They're the undisputed masters of genetic optimization, having evolved under immense pressure to do more with less. And this leaves us with a really fascinating thought. As we develop incredible technologies like gene therapy, where we need to deliver and express genes as efficiently as possible, could we learn from the viral playbook? Could their ruthless, ingenious strategies for optimization one day be harnessed to rewrite medicine itself?